Good afternoon, everybody, and welcome to our September virtual masterclass on ensuring accurate classification of variants in BRCA1, BRCA2, and other HRR genes. Next slide, please. So normal housekeeping rules apply to this webinar. So we've automatically muted your microphones to try and avoid any background noise. That will be of benefit to you if you're unable to watch it live, because we are recording this webinar and it will be made available to you through our EMQN and GenQA YouTube channels, probably 10 days to 2 weeks from this date. But most importantly, this is an opportunity for you to learn about the subject we're going to be talking today. We use the Q&A function to allow you to submit questions to us. We have a dedicated session at the end of it where you can pose your questions to the experts. This is a fundamental part of what we're trying to do. We're providing an educational resource here, so please do submit your questions. If we don't get around to answering live on this webinar, then we will, of course, do our normal, which is pick them up afterwards and get back to you with some feedback. Okay, next slide, please. That leads me very nicely on to feedback itself. So we also listen to what you're having to say in terms of what your feedback on this resource and this opportunity. So please do feedback to us afterwards. You will receive a link in a short email after the webinar. Takes 2 or 3 minutes to complete. Strongly encourage you, please, to feedback to us and to help us improve and move forward for next year's webinars. With that, I'm going to hand over to my co-chair, Professor Sandi Deans, for the introduction.
Thank you very much, Simon. So great pleasure to introduce our very exciting webinar today. We have a huge amount of detail to share with you, and that is coming from our panel of experts. So today we have Miranda Durkie from Sheffield in the UK. We have Alice Garrett from London in the UK, and also Kathleen from Ghent in Belgium. So a great big thank you to you all for being here today to present some of the great detail around the variant classifications for these runs. So next slide, please. So this is a disclaimer for all of us. And then next slide, please. We move on to the agenda. So we're going to get the CanVIG-UK recommendations for classification of reduced penetrance variants in high penetrance cancer susceptibility genes, interpretation of Splice variants in HRR genes, and then we're also going to then close with the review of the learning from run 12 EQA. So that's the Genie module for classification of variants. Now we also, as Simon mentioned, have the Q&A at the end of the webinar. So please do put your questions in and we'll hope to be able to answer some of them. So I'm now going to hand over to Alice, who is going to give us an update on the CanVIG recommendation. So Alice, over to you, please.
Thanks very much, Sandy. So good afternoon, everybody. My name's Alice Garrett. I'm a clinical lecturer at the Institute of Cancer Research, where I do a lot of work with CanVIG-UK, which I'll talk about in a bit. And I'm also a locum consultant in clinical genetics at St George's Hospital in London in the UK. Next slide, please. So just before I kind of get going with talking about reduced penetrance variants and how we're choosing to approach this in CanVIG-UK, I'll just give a little bit of background about reduced penetrance in general for those of you who haven't kind of thought about it much before. So when we are doing testing for patients and giving results about likely pathogenic or pathogenic variants in cancer susceptibility genes, we tend to give penetrance estimates, which are a sort of average of what the penetrance seems to be based on our observations of all pathogenic or likely pathogenic variants in a given gene. But this is almost certainly pretty simplistic, and actually the true penetrance probably varies quite considerably between individual variants. And actually it might vary differently for different phenotypes that are associated with a given gene. Next slide, please. So an example of this that we do know about is the breast cancer and ovarian cancer cluster regions that we see in BRCA1 and BRCA2. So we know that certain regions of the gene, you're likely to have a higher risk of ovarian cancer. In other regions, you might have a higher risk of breast cancer. Next slide, please. Oh, okay. Oh, no, not at the same time, please. So yeah, so the kind of poster child of reduced penetrance is this BRCA1 R1699Q variant, which those of you who've worked a lot with BRCA1 will probably be familiar with. And some excellent work done by the Enigma Consortium, collating data from lots of different families, demonstrated that this variant has quite considerably reduced penetrance compared to a standard BRCA1 variant. And they follow this up with a subsequent publication in 2018, where they did some more analysis and also gave some clinical recommendations about how to manage carriers of this variant. So if you just put the next animation, please. And again, please. Thank you. Super. Yeah. So if you just look at the figure on the left, this shows their sort of estimates of penetrance based on the data. So the red line represents the cumulative risk for carriers of standard penetrance BRCA1 variants, which you can see is very high for breast cancer and ovarian cancer. The green line is the population cumulative risk, and you can see this variant represented by the blue line is somewhere in the middle, both for breast cancer and for ovarian cancer. Next slide, please. And so the Enigma team gave some clinical recommendations, which were taken up by the UK Cancer Genetics Group. So they essentially say that if somebody is found to have this variant, they should start their mammograms slightly later than usual, so at 40 rather than 30, could potentially delay having salpingo-oophorectomy, and also risk-reducing mastectomy may not be appropriate as it would be for carriers of full penetrance BRCA1 variants. Next animation, please. And CanVIG-UK has developed a report template for how to report this variant, which was actually developed by Miranda, who's you'll be hearing from next. Next slide, please. So again, this is just sort of an overview of what I've talked about already, which is a sort of schematic developed by the ClinGen group, and it divides variants loosely into these three categories, although it is a kind of continuous spectrum of penetrance. Next slide, please. So on the right hand side, oh, just go back, thank you. Yeah, on the right, there are these high penetrance variants, which we're pretty familiar with. This is what we use our standard Richards et al. ACMG variant classification framework for. Next slide, please. And then on the bottom end, we've got these risk alleles, which are variants which are associated with some risk of disease, but probably quite a small risk. And they might, for example, be used in polygenic risk scores. And there is a ClinGen low penetrance risk allele working group who have developed some recommendations for how to report these. Next slide, please. So CanVIG-UK decided to work on this middle group. It was called low penetrance by ClinGen, but we've actually designated it as reduced penetrance, because if something goes from high penetrance to slightly less high, it might still be kind of moderate risk rather than sort of low risk. Next slide, please. And yeah, just before I go on, I'll just introduce CanVIG-UK for those of you who aren't familiar with it. So this is a group of almost 400 members at last count, representing labs and clinical genetics services across the UK and the Republic of Ireland. It's been going since 2017. We have monthly meetings and where we discuss various issues relating to variant interpretation in cancer susceptibility genes, and have tricky cases and also development of consensus recommendations for the UK and the Republic of Ireland. We also host the CanVIG-UK platform and email forum, which if you haven't looked at it, I'd strongly recommend you go and have a look. It has lots of useful information that can help you with variant interpretation. Also this email forum that allows you to communicate with other labs within the UK and internationally about tricky variants. And that pie chart at the bottom just shows that approximately half of CanVIG members are clinical scientists and the rest are mostly clinical geneticists, but there are people with other backgrounds as well. Next slide, please. Or next animation. Thank you. Yeah. So we also have the wonderful CanVIG Steering and Advisory Group, CSTAG, of which Miranda and others across the UK and Republic of Ireland are members, which is a sort of smaller working group which meets between CanVIG meetings to develop guidance and drive things forward. And the photograph there is just of Sophie Allen, who does a fantastic job of coordinating the efforts of both CanVIG and CSTAG. Next slide, please. So this is just a sort of timeline of the development of the reduced penetrance framework, which I'll take you through. So this all started back in, well, pretty much exactly a year ago, actually, when we sent a poll round to CanVIG members to sort of see what the current situation was. Next slide, please. And again. So these are the results of the poll. So we were sort of asking how often people were encountering variants that they thought might be of reduced penetrance. And there was some variability in responses, but the most frequent response was 2 to 6 monthly. Next slide, please. And about half of respondents said that they had previously classified a variant as being of reduced penetrance locally. Next slide, please. Thank you. And then this question was about how confident people felt about classifying variants as being of reduced penetrance. And when classifying them as an individual without any external discussion, people in general didn't feel very confident. If they discussed it regionally or locally, so that's the GLH is the Genomic Laboratory Hub, they felt a bit more confident. But if they discussed it nationally, for example, at a CanVIG meeting, in general, they felt a lot more confident. Next slide, please. And then we asked people a slightly leading question of how useful they thought it would be to develop a framework. And everybody said extremely valuable or very valuable. Not entirely sure of the difference between those two things, but essentially they were very keen that we produce something to help them improve consistency. Next slide, please. So then in the next follow-up CanVIG meeting, we did a live survey on some exemplar variants to kind of, or hypothetical situations to see what people's thoughts were. Next slide, please. So this is the results of some questions about we asked about different scenarios using case control evidence. And just to kind of orientate you, when we're talking about high penetrance cancer susceptibility genes, we're usually talking of an odds ratio of sort of 4 or above. So for this reduced penetrance range, we were more looking at sort of a range of 2 to 4 rather, for the odds ratio. So we sort of played, don't worry too much about the exact figures, but we kind of played around with different point estimates and confidence intervals for the odds ratio to see what people's thoughts were. And the summary is that there was quite a lot of variability in people's responses and how they would assess this evidence, which sort of highlights the need for a more consistent approach. Next slide, please. And again, we looked at scenarios using functional data. So, for example, functional assays giving conflicting results or results in the intermediate range. And again, there was some variability in responses. Next slide, please. And then we also looked at some scenarios that also could indicate possible reduced penetrance. For example, an observation of the variant in a homozygous state, or in chance with a pathogenic variant in somebody who has either no phenotype or a weaker phenotype than would normally be expected in that situation. For example, minimal features of Fanconi anemia in somebody who's homozygous for a BRCA2 variant. Just go back one, please. Yeah, and also segregation data towards benignity under a full penetrance model. Next slide, please. So we then set about with CSTAG drafting a reduced penetrance framework. Next slide, please. And so this was our first draft. And just to kind of highlight that we divided the situations into where we have active evidence that there appears to be a reduced penetrance effect size, most likely from case control data, but potentially also from segregation analyses. Next slide, please. And then the other side is where we don't actually have quantifiable evidence indicating an effect size, but we're suspicious because, there's conflicting clinical data or a very weak phenotype, for example. Next slide, please. So we circulated this draft framework to CanVIG-UK members, and then presented some exemplar variants and did a survey of how people thought these should be classified and the how they might apply this framework. Next slide, please. And essentially, the variants where there was quantified evidence of an effect size, the majority of people were happy to classify as likely pathogenic with reduced penetrance. So those are the pie charts in the middle and left. As you can see from the pie chart on the right, where there was no active evidence of reduced effect size, but there was a suspicion from weakly pathogenic or potentially conflicting evidence, people thought that it should still be classified as a variant of uncertain significance. Next slide, please. And that was replicated when we asked people's confidence and how comfortable they felt with giving a classification of reduced penetrance in these different scenarios. So where there was either quantified case control evidence or quantified segregation evidence indicating a reduced penetrance effect, people were generally happy to classify as such, but without this, the majority of people said they wouldn't be comfortable. So this was clearly the area that we needed to go back and have another look at. Next slide, please. So we went back to Paul Seestag and adapted the draft framework. Next slide, please. Particularly working, as I said, on that area where we don't have the active evidence of an effect size, and just added a few more kind of caveats and areas of caution to make sure we weren't being overly lenient here and weren't just sort of ignoring evidence we didn't like. Next slide, please. So we then trialed this draft framework, this updated version on some test variants, and again went back to CanVIG members at a meeting to survey them on their thoughts on this. Next slide, please. And so when there was substantial evidence towards pathogenicity, but a normal homozygote reported in the literature, the majority of people were happy that this could be classified as likely pathogenic with reduced penetrance, although there was definitely not a kind of unanimous agreement, which sort of exemplifies how tricky this area is and difficult to kind of reach consensus. Then we had another couple of test variants where there was some evidence towards pathogenicity, but some segregation or family history data towards benignity under a full penetrance model, and people thought that should probably still be a VUS. But there was substantial evidence towards pathogenicity, but just a bit of segregation or family history data towards benignity under a full penetrance model. The majority of people were happy to classify that variant as likely pathogenic with reduced likely pathogenic with reduced penetrance, which is because if, this data has been calculated under a full penetrance model and the variant is truly got reduced penetrance, we wouldn't expect it to behave the same way as a variant that did have that full penetrance based on the clinical observations. Next slide, please. So we finalized this framework in July and circulated it to the CanVIG membership. So next slide, please. So you've already seen quite a lot of tastes of this throughout the presentation, but I'll just take you through the structure again. So on the left-hand side, as I say, is where we've got active evidence of a reduced penetrance effect size. And this usually comes from the case control data, and we've set out the sort of criteria you need to reach for that. And also, if you've got some segregation data, for example, from the Cool Bayes tool, how you might incorporate that. Next slide, please. There's a number of different evidence criteria that can be used at standard strength, so as per the normal full penetrance guidance. Next slide, please. And then for the functional data, so that's PS3, and for observation in a homozygous state or in trans with a pathogenic variant, So that's PM3 for those two criteria. We've got some additional stipulations for how you might be able to apply this. For example, if there's intermediate functional result on an assay or a mild phenotype, that could still count as evidence towards pathogenicity in a reduced penetrance context. Next slide, please. And then we've also got some recommendations for how to deal with evidence that would normally point towards benignity. So in some situations, it might be appropriate to discount that or at least downgrade it by an evidence strength. So it doesn't prevent you from giving a classification of likely penetrance with likely pathogenic with reduced penetrance. And that's that varies a little bit depending on whether you start with that active evidence or not. And next slide, please. And then we also give recommendations about how to combine all of the evidence to give an overall classification, which again varies a little bit between the two different scenarios. And you'll see from the final framework that the more we looked into this, the more complicated it got. So there is quite a long list of additional notes and caveats in the final version. Next slide, please. So just to summarise, these variants, which are suspected to be of reduced penetrance, are relatively frequently encountered by clinical diagnostic laboratories, and it's probably not appropriate to use the standard full penetrance variant interpretation framework, and a lot of labs have historically been struggling with this. And this is why we've been seeing that these classifications might be inconsistent between different labs or over time, and they might flip back and forward between variant of uncertain significance and likely pathogenic. And we all know the challenges that come with that for all concerned, the clinicians, the patients, and the scientists as well. Next slide, please. Everyone that we surveyed was very keen that we develop some pragmatic guidance to help try and improve or start to improve consistency, knowing that this was not the perfect solution. So we've developed this framework within CanVIG-UK, and we use BRCA1 and BRCA2 variants only for the development of this framework. But in theory, a lot of the principles could apply to other genes, and we just haven't tested it on those yet. And we looked at scenarios where there was quantified evidence of a reduced effect size from case control or segregation data, and where there wasn't. Next slide, please. And of course, reporting these variants as being likely pathogenic with reduced penetrance is all very well, but we do need concomitant clinical guidance so we actually know what to do with these patients and how to manage them. That guidance, as I said, does exist for BRCA1 R169Q, but that doesn't mean that can automatically transfer to other variants, and there are challenges of integration of tools such as CanRisk as well. So we're working on it. And just a huge thanks to everybody who's been involved. So all the members of CanVIG-UK and all the people who've responded to the numerous polls and surveys. And of course, CSTAG members who've worked very hard on this, and the team at the ICR, in particular Claire Turnbull and Sophie Allen, and also the funders of the CanGene CanVAR programme, which has funded many of the people working on this, and they've also funded the CG-MAVE programme, which has just started. Thank you very much. And I'll just hand over now to Miranda, who I think is going to talk about splicing variants.
Yeah. Hi, everybody. Thank you very much, Alice. That was excellent. So I'm Miranda Durkie. I'm a senior clinical scientist in Sheffield. Next slide, please. Sarah, I've got quite a lot of animations, so I might just say next if that's okay. I'm being rude. So yeah, I am going to cover splicing in HRR genes. So I'm going to start with a very quick overview of splicing. Then I'm going to spend the majority of my time talking about the ClinGen SVI splicing subgroup guidelines. So these were published by Walker et al. Last year. Then I'm going to go into a bit more detail about some of the guidelines that we've got available for specific HRR genes. I feel like at this point, I should say a bit of a caveat, because as Alice has said, I've been involved in the writing of the CanVIG guidelines, but not with the Enigma, ATM or the Walker et al. Guidelines. So it's very much me as a clinical scientist reviewing these guidelines. Thank you. Next slide, please. So we're going to start with the overview of splicing. I'm afraid, because of this, the way the slides are being shared, I don't have a pointer. But I'm hoping that most people are kind of aware that splicing, the most important sequences are at the 3' end of the exon. You've got your GT at the +1, +2 position, and that donor splice site and dinucleotide, and then on the on the right hand side at the 3' end of the exon for your acceptor splice site, the -1 and -2 position where you have that AG site. And for the purposes of the rest of the talk, I'm going to call these the canonical splice sites. There are a number of of other really important splicing nucleotides that affect splicing. So that's including the A at the branch point, and then a number of both exonic and intronic splicing enhancers and silencers. Next, please. And this is maybe a little bit small, but I also wanted to highlight, and if you can see the 5' splice site labeled on this, and actually the last nucleotide of the exon, where that's a G, that is also very important for splicing, and the +5G is also really important for splicing. So it's not just those dinucleotide canonical splice sites. And most of the time, we are looking at variants that are affecting the major spliceosome. And over 98% of the splice sites are these GTAG, but we actually have about 1% of still major spliceosome splice sites that use a GCA to G. Next, please. And this means is very important because it means we have a subset of these +2 T to C variants that can actually be functional. And in order to kind of work out which ones those are, we set a very high threshold of your splicing in silico tools. And so even if you have got a canonical splice site, it is important to check your splicing in silico tools. It also means that we have a rare number of these +2 C to Ts that actually just increase the efficiency of splicing, but you still have completely normal splicing. So these will also be functional. Next, please. Then you often hear a lot talked about the minor spliceosome, but in fact, these are very, very rare. And these are only used in less than 500 genes across the genome. So it only affects about 0.1% of splice sites, but they are AT-AC. And the reason I'm kind of highlighting this is that the splicing tools are not quite as accurate for predicting whether you get splicing at one of these splice sites compared to the major spliceosome. Next, please. Okay, so what happens if you get a variant that is affecting your canonical dinucleotide splicing? So if we look at the one where you've got the kind of little red dots either side of exon 2, if you have a variant that's affecting either of those canonical splice sites, the most common and most likely event you will get is exon skipping. Next line down, intron inclusion. This doesn't occur very often. It's usually only when it's a very small intron, particularly if it's a large intron, that doesn't tend to occur. And then kind of at the bottom is shown that if you're getting rid of your canonical dinucleotide splice sites, that you can get use of a cryptic splice site, either within the exon or within the intron, and then creating a range of different effects. In addition, what's not so shown in this diagram is if you have a variant at a non-canonical position, so potentially it's in the middle of one of the exons or in the middle of an intron, and obviously this can create a new cryptic splice site. And really, really importantly, this is a bit of a theme I'm coming back to, is that Mother Nature is not just black and white. You don't always just get one event, but you can get a combination of different effects as well. Next, please. So now looking at the ClinGen SBI, and they start the paper with this really lovely graphical abstract showing all the different ACMG codes that you can use for splicing. All the ones in red are towards pathogenicity, and all the ones in blue are towards benignity. Next, please. And what the authors have tried to do is to use these guidelines as a framework to help the expert groups, particularly the VSIPs, but if there isn't VSIP guidelines available for anyone looking at a gene where there isn't that gene-specific information available, to work out how, what strengths of PVS1 and other splicing codes you can use. So this diagram is a kind of a hypothetical gene showing some of the most common scenarios that you could see if you were looking at your canonical splice sites. Next, please. So let's start here. If you've got a splice variant that's affecting an untranslated upstream exon, it's not affecting the coding sequence, then PVS1 would not be applicable. Next, please. In the next scenario, you have got a splicing variant that is eliminating your ATG initiation site. Very importantly, you need to check if there's not another alternative ATG start in the next exon. But if there's not, then you can use PVS1 at full strength, very strong. Next, please. This next one's very simple. You've got an outer frame transcript, it's going to be subject to nonsense-mediated decay, and you can apply PVS1 at maximum strength. Next, please. I've grouped the next two together. So exon 4 and 7 are both in frame, let's say deletions. And in both cases, they're not affecting a critical domain. They may be affecting a domain, but the role can be uncertain. And the strength that you assign to PVS1 is very much dependent on the size of the protein that is, or coding sequence that is affected. So in the case of exon 4, that's affecting greater than 10% of the coding sequence. So you can give that a strong, whereas for exon 7, it's a smaller deletion. So you're going to give that a maximum of moderate. Next, please. Again, I've grouped these two together. So these are, let's say, exon skipping affecting a critical domain. For exon 5, it's very simple. It's outer frame, NMD, etc. So we would be very happy using that at PVS1 very strong. For exon 6, according to the previous PVS1 decision tree by Taeun et al., if you had an in-frame deletion affecting a critical domain, you could upgrade that to strong. But these authors are actually recommending that we can upgrade that again to very strong, which I think is very helpful and useful, and why I've put that red star there. Next, please. And then finally, if you have a splicing variant affecting the 3' end of the gene, it's not affecting nonsense-mediated decay, you're only losing a small amount of the coding sequence, then you are having that capped at moderate. But the idea is to then kind of convert this to whatever your gene of interest is. Next, please. The authors also did a huge amount of work on the in silico tools. I'm just showing kind of a fraction here, but it was quite nice because they compared 11 in silico tools using the Greg Finlay's saturation genome editing assay that was done on BRCA1 variants, so very relevant for this group. And using these 414 variants, where we know the functional effect, they found that Splice AI has the highest area under a curve. Next, please. So just a reminder that we cannot use the PP3, the in silico tools, with PVS1. Next, please. And it was very reassuring and pleasing that the authors did a lot of work at setting the boundaries and what the cutoffs should be for the use of the Splice AA delta score for PP3 and BP4. And actually, this was completely consistent with some other papers that have been published and have the same as the CanVIG guidelines have used for a number of years. In addition, they allow the use of BP7 for either synonymous or intronic variants, but making sure that you're avoiding that kind of really spliceogenic region around the splice sites. Next, please. The next code that they have looked at and has been previously used in other guidelines, but they've kind of expanded the use of, is PS1. And so this would be for a variant with the same predicted splicing event as a known pathogenic or likely pathogenic variant. So the prerequisite is that the predicted event of the variant under observation must precisely match the predicted event of that pathogenic variant. Next, please. And if you can see kind of on the top line, you can use this code for both the canonical and the non-canonical variants. And looking at the top line, If you have a variant affecting the same nucleotide and it's a pathogenic variant, then you can apply PS1 at its kind of top strength of strong. If it's a lightly pathogenic variant, you would downgrade by 1 level of strength. And then you can also see where I've highlighted in yellow, you can also now use PS1 for other variants within the same region, bearing in mind it's still got to have the same effect. You can't compare one variant predicting a loss of, say, a donor site with another one that's predicting the creation of a new cryptic splice site. And so those ones within the same region are downgraded 1 from level from the same nucleotide. Next, please. And as is the direction of travel for all of the new ACMG guidelines, they develop these very comprehensive, very, very helpful decision trees. It's very big, their decision trees. This is just part of it. Next, please. So we're just going to stick with the theme of splicing data only. Next, please. So if we look first at the non-canonical splice sites. Next, please. And again, please. So in this example, we're assuming that we've looked at splicing, we can apply PP3, and then you're going to look for other variants at that nucleotide or in that region, and then apply your PP3 along with the appropriate code of PS1. Next, please. So worth mentioning that if you are applying BP4, you don't get to apply PS1. So now moving on to the donor, the canonical splice sites. So if we go back, if we think about what the flowchart that I first showed was, that you're going to apply PVS1, but you really need to work out what the effect of that variant is going to be on the coding sequence. So you want to look whether it's out of frame, in frame, is it affecting a critical functional domain? So you're going to kind of decide on your strength of PVS1 from that. And the next, please. When you have that code, you're then going to look for other variants within that region and apply the appropriate strength. You'll notice that if you are applying your maximum strength of PVS1 very strong, then the PS1 is capped at supporting. But for the reduced strengths of PVS1, then you can use the higher strengths of PS1. Next, please. The next area that they looked at is the use of RNA assay data. And from the Richards et al. Original ACMG, this all fell under PS3 and BS3. But these authors have recommended that we continue using those codes only for well-established functional assays of protein effect. And instead, for RNA splicing assays, we should use PVS1 RNA and BP7 RNA at variable strength. Next, please. So looking at another area of the decision tree, next please. The first thing that it says when you've got some RNA and splicing data is that you need to consider the quality of that assay. So looking at your the technique, the primers used, the RNA source, etc. So you're really using your scientific and professional judgment to say how good a quality is this assay. Next, please. Following again the decision tree, if we have no variant-specific observed impact and it's an intronic variant, we can apply BP7 at strong. Interestingly, the authors have also allowed us to keep our prediction scores, whether that be PP3 or BP4, as well as that BP7. Next, please. And there's a big caveat here. So if your variant is actually affecting the coding sequence, so for example, it's a missense change, before you apply BP7 at strong, because it's a lot of evidence towards benignity, you really need to exclude that that variant is not having an effect on the protein. Next, please. Okay, so now we have an RNA splicing assay. We can see a variant-specific effect. We've considered it's a good assay. Again, you're going to follow your flow chart to work out the strength of PVS1 that you are going to apply. Next, please. And then you need to do another assessment of that RNA assay data. And it's really looking at the quantitation of the amount of non-functional or aberrant transcript produced compared to the amount of either wild type or other functional transcript that is produced by that assay. If it's complete, so nearly all of your variant is completing this aberrant loss of function transcript, then you can keep the same strength of PVS1 that you've applied from the decision tree. If it's near complete, but you can see some wild type there, you can reduce the strength by 1 level. But if it's really incomplete, then you would not apply the codes. Next, please. So that's really important when you're thinking about this RNA assay data, that you're really looking at the combined analysis and thinking about the assay PVS1 decision tree before applying your weight. Next, please. And so I thought it'd be helpful just to show you what I mean by kind of that complete or incomplete, or we also refer to it, particularly in the CanVIG guidelines, as leaky splicing. So this is a fantastic paper by Nix et al. I'm going to use a couple of examples from this paper. I know there's quite a lot going on this slide. So if you just look at the 2 red boxes, you can see that this is a variant C.68 - 3T to G, and we would predict a skipping of exon 3. You can see that on the gel, but you can also see that there's a lot of the wild type normal transcript produced, and that's kind of shown in the box below. And the bottom right shows you that the variant is actually sitting very much with the benign controls in that family history weighting. Next, please. Another fantastic paper, and again, I'm going to come back to other examples from this one. This is quite a complex area around exon 12, BRCA2, but I've highlighted in red 4 different variants, and then I've highlighted in yellow the full length wild type transcript. And you can see for all 4 of these variants, they are producing some wild type transcript at very variable levels, plus some alternative different transcripts. And so it's really important, as you can see from that, that we quantitate the amount of that wild type transcript and that aberrant transcript produced. Next, please. So the paper talks a lot about rescue mechanisms, and this is in general, not just kind of thinking about splicing, but we have 4 common rescue mechanisms that may affect an impact of loss of function variants. The first is a protein truncating codon at the 3' end of the coding sequence, which is not subject to NMD. You can get a shorter functional protein. Next is a stop gain in or exon skipping of a non-constitutive exon. And by that, I mean an exon, they're usually in frame, they're usually naturally occurring in alternative transcripts where that exon is skipped, and they're not affecting a critical domain of that gene. Next, please. You can also get in-frame deletions or duplications that, again, aren't subject to NMD that can create a functional protein. And finally, you can get rescue of initiation codon variants by use of an alternative in-frame ATG. Next, please. So there's a lot more information in the guidelines, in the supplementary data. And again, they've set it out so that the expert groups can define this for their gene of interest. So in this example, this is an in-frame splicing event. And the next step would be to quantify the relative expression of your transcript, then to look at whether it's affecting a critical domain, then to look at what the splicing event is, what impact that's going to have on the protein sequence, to identify whether you get a rescue transcript. So this is just a framework really to determine what candidate rescue transcripts are available for your gene of interest. Next, please. And so this is a kind of a worked example. So if we look at the stop gain 2 on the left-hand side, actually it's very kind of similar, so I might as well do both together. On the right-hand side, we've got a splicing variant affecting a -1 position. And you can see in the kind of grayed out exons that in both cases, you have an alternative transcript that on the left-hand side is skipping that exon 3, on the right-hand side is skipping that exons 2 and 3. So if you have that stop gain or that splice variant, that you can get upregulation of those naturally occurring in-frame transcripts that can rescue it. Next, please. I'm going to give you a real-life example of this. So this is a BRCA2 variant. It's a missense variant, but it actually affects the last nucleotide of exon 4. The Revel score is very low, and it is not affecting a critical functional domain, so it's unlikely to have an effect on the protein via the missense route. But the Splice AI delta score is very high, 0.97. So we predict a skipping of exon 4, which would be out of frame. Next, please. So this variant was widely reported as likely pathogenic until, again, coming back to that Mesman and the Nix papers, did a lot of work on this variant. And again, there's quite a lot going on, but if you look at this red box and you remember back to one of my first slides where I said there could be a combination of different events going on. So you can see the deletion of the 4, the delta 4 is there as we would predict, but there's a huge host of other transcripts and other alternative splice variants being produced by that one variant. Next, please. And I've highlighted the del45 and the del47 because actually both of those are in frame. They're not affecting a functional domain. They're naturally occurring, and it's been shown to have normal HDR activity. So this is a rescue mechanism. And just to labour this point a little bit more, so we haven't actually seen this variant in the UK, but we have 13 cases with C.425G to A. Also a very weak missense change. We had previously done RNA studies, confirmed a deletion of exon 4, and this had been widely reported as likely pathogenic. On the basis of this data coming to light, we've now repeated that RNA studies and can now see that there is that del45 and del4-7, and that variant has now unfortunately had to be downgraded from likely pathogenic to VUS in 13 families. There's been multiple predictive tests, so yeah, you can imagine the anxiety and stress for everyone kind of involved. Next slide, please. So a bit of an expert tip here. I've seen it in this paper. Next, please. But actually, it's been quite widely reported in other, and there are some in silico tools that kind of automatically do this for you. If you're using Splice AI lookup, then the default distance is normally set at 50 base pairs either side of the variant. But actually, if you can expand that to 5,000 base pairs or even 10,000 base pairs, then you can see your concordance with your RNA-seq analysis and percentage misclassification rate is actually significantly better than just using Splice AI with the default. So it helps you to see that alternative splicing, and it can help to see if you've got the pseudo exon activation as well. Next, please. Next, please. This is all in the supplementary data. I haven't got time to kind of go through it all, but please have a look because it is really important that you need to take into account when you're assessing these RNA assays. Next, please. So the key takeaways in this paper is that we can now use PVS1 very strong for in-frame events affecting those critical functional domains. Next, please. They've done a lot of work on the in silico tools. Next, please. We can also use BP7 for synonymous variants where BP4 is also met, but as I said, avoiding those kind of regions that are most spiciogenic. Next, please. We can now use PS1 for similar splice variants within the same region. Next, please. And as I said, we're reusing the PVS1 RNA and BP7 RNA codes at variable strengths for our splicing assays. Next, please. Remember to consider your rescue isoforms. Next, please. And the authors have really defined a process to develop gene-specific PVS1 decision trees. So this brings me really nicely on to the next slide, please. The Enigma guidelines. And the really good news is that Enigma and the ATM VSET work really closely with the splicing SBI to make sure all of their codes and all of their guidance is fully compatible. So Enigma have developed their PVS1 gene-specific decision tree. It's shown there on the left for the canonical splice sites. They've also gone a step further by listing every single splice site that you could possibly get in either gene and which strength of PVS1 that you can use for it. You can see kind of a couple on the right-hand side that are labeled green, where they've downgraded kind of from the recommended strength in Walker et al. And all the reasons why is all explained in the supplementary information. Next, please. So that's for canonical splice sites. If you have a non-canonical BRCA1 or BRCA2 variant, you would follow this figure 9 decision tree. So you're going to start by looking at your bioinformatics code. So let's assume it's an intronic variant where you can see a predicted impact on splicing. You're going to apply PP3 and then you're going to look for your PS1 splicing, just as we did with Walker et al. Next, please. Next, you're going to have a look for your splicing assay data. Next, please. And just the same as the Walker guidelines, you're going to review your assay design and quality, and then you're going to use your decision tree to work out what strength of PVS1 you can apply. Next, please. And then the next step, as we did previously, is to review your data and look at the quantitation of your percentage of wild type versus aberrant transcript produced. And Enigma have very kindly put this all in a very nice table, table 9. Next, please. So this table is to be used for the weighting of RNA data for non-canonical splice sites. And they've split it into kind of 2 sections. At the top, you've got assay results with quantitation, and at the bottom, assay results without quantitation. Next, please. So let's have a look, first of all, where you've got an assay, let's say a patient with quantification, where more than 90% of the transcript produced is non-functional. So I should note, point out that in the blue, you're saying you've got less than 10% wild type or assumed functional. So in the example that we were looking at before, the DEL45 or the DEL47 would fit into this blue group. It would be assumed functional. So where, you have it's nearly all aberrant transcript, you can apply PVS1 for your patient at exactly the same strength as you have got it from your PVS1 decision tree. However, if you have a mini gene assay, because these are only hemizygous, you haven't got all of the other splicing factors and proteins kind of in there as you would do in a real patient, you are going to downweight everything by 1 level of evidence, because we do see some differences in the mini gene assays. Next, please. So now we're going to have a look at example where you've got between 80 to 90% is aberrant, between 10 to 20% is wild type. Again, the patient, you're going to downgrade 1 level from your PVS1 decision tree, and then the mini gene assay is downgraded 1 level below that. Next, please. And this is what's really, really helpful for the BRCA1 and BRCA2 genes. So Enigma have defined that if you have 7, well, actually it's easy to look at it in terms of the functional transcript. If you have 20 to 30% functional transcript, you should not apply PVS1 RNA. So that would be not applicable. And actually, if you have 30% of wild type or functional transcript, you would actually be able to apply BP7 at strong. Next, please. If you have an RNA assay, but they haven't been able to perform quantitation, then you would just be looking to see whether it's near complete splicing. If it looks like that, then you're going to downgrade the highest strength to strong, and you're going to use moderate for all the others. If it's incomplete, then you're just not going to be able to apply your PVS1 RNA score. But if it looks like it's pretty much the same as all your controls, then you're going to be able to apply BP7 strong. Next, please. And then I've just got this one slide on the ATM guidelines. And what I really like about the ATM guidelines, they've again followed the Walker et al. They've got the same decision tree, but as well as including the canonical splice sites, they've also added in that last nucleotide of the exon where it's a G to a non-G. And they've given a few more kind of caveats. You have to check the adjacent sequence, and as long as it's not that, then you can use whatever the PVS1 strength is there, but downweighted by 1 level of evidence. Next, please. And then we talked kind of right at the beginning about the +2 T to C variants. They have got plenty of these on their very strong list, but you can see for these 5 variants that they have decided that there could be normal splicing produced from these, so PVS1 would not be applicable. And they have got 1 +2 C to T variant that improves the donor site that we actually included in, I think it was 2 runs ago, so it's in run 11, and unfortunately did catch quite a few participants out. Next slide, please. And then just quickly looking at the CanVIG guidelines. Next, please. You can see that we are still at the moment under the version 2.19, scoring this data under PS3. In a lot of ways, it's just semantics, as we've always allowed the use of the strength up to very strong if you have two different assays with no evidence of leakiness. Next, please. And we have a lot of information about the assay design, the number of controls, and the leakiness, etc. Next, please. But I've decided not to dwell on these guidelines because we do have this draft version 3.0, which should hopefully be out soon, where we have aligned with the Walker et al. Guidelines and moved everything to PBS1 RNA, hopefully made it all a bit clearer and aligned the leakiness to that same percentages. I should say that these guidelines will not be available for use for Run 13. So anyone that's doing Run 13, please continue to use the version 2.9. And that was a very whistle-top tour of splicing. I'm now going to hand over to Kathleen.
Thank you, Miranda, for this nice overview of a difficult topic. So I'm Kathleen Klaas from the Center of Medical Genetics, where I'm working as clinical laboratory geneticist in Ghent, in Belgium. So next slide, please. I will give you an overview of the take-home messages from run 12, so the previous run. And the aim is that I highlight some of the parameters where there were some difficulties with, and I will try to illustrate it with examples and try to refer to the guidelines that are currently being used. So in run 12, there were 6 variants that had to be classified, and you see an overview here. And there were only 7 participants out of 318 that completed all variants who successfully classified all 6 variants. However, and next please, if we look in more detail, you can also bundle the variants that have the similar clinical impact according to pathogenicity, meaning the clinical impact of class 1 and class 2 and of class 4 and 5 are similar. So if we look then in this way to the outcome, then we see that the large majority had correct classifications for 5 out of 6 variants, only for class for the third variant, which is the BRCA2 Val 391 ILE variant, was classified by several persons as class 3, while it was actually a class 2. But let's dig into the parameters where there are sometimes differences between CanVIG and Enigma, and some of the parameters that caused some aberrations. So next, please. So the first parameter I would like to highlight is the PM2 parameter. This is the parameter about absence from controls. And there is a difference in strength between Enigma and CanVIG. So Enigma applies it a maximum at a supporting level in agreement with the ClinGen SVI recommendation that was published back in 2020. So where they said that when a variant is absent from controls, it should be maximum used at supporting, because actually being a rare variant is quite frequent. So there are many variants there that are only reported once in this control population. For CanVIG, you can use it at moderate when there are no observations of the variant across all populations in GNOMAD version 4.1. So they are already using the most recent version from GNOMAD. And then at supporting level, if the variant is not absent, but if it's present in less than 1 in 50,000 individuals or less than 1 in 100,000 alleles. There is one caution: if you have already applied the UK Biobank to use it for the PS4 application, then you should use in GNOMAD the non-UK biobank partition, because otherwise you will do double counting. Next, please. So the next parameter I would like to discuss is PVS1. So this is about the null variants, including the nonsense frame shifts variants, initiation codon, and single and or multi-exon deletions in a gene where loss of function is a known mechanism of disease. And this is of course the case for the variants we are discussing now. I will skip the part on the canonical splice sites and the other splice sites because Miranda has explained this already very nicely. In the Enigma guidelines, you have decision trees for BRCA1 and BRCA2. Your next slide, please. And here you see the relevant part of this decision tree. So for both genes, you can see that the initiation for the initiation stop codon, if there is a variant there is no known alternative start codon known, so you can use PVS1 at its full strength. For protein truncating variants for which NMD is predicted, you can use it in BRCA1 at PVS1 if the variants are not present in exons 8 or 9, or if you use the historical numbering, these are exons 9 and 10. Because if truncating variants are present in these exons of BRCA1, PVS1 should not be applied. Then the protein truncating variants for which nonsense mediated decay is not predicted, you can really apply PVS1 until almost the very end of BRCA1, because at the end there is also critical, domains that are critical for the protein function. However, if it's in a region of which the protein function is unknown, then PVS1 should not be applied. For BRCA2, you cannot apply PVS1 in case the role of the truncated or altered region in the protein function is unknown. But please note that the coding, the last truncating variant that's known to be pathogenic is at codon 3309 in BRCA2. Next, please. What's then the difference with CanVIG? Well, for CanVIG, the guidelines are based on the decision tree that is published by Tayoun et al. And you can see the relevant part of it here at the bottom of the slide. Please note that for in-frame insertions of deletions, you should use PM4 instead of PVS1. And PM4 is that the protein length changes as a result of in-frame deletions or insertions in a non-repeat region. The big difference is then that Enigma and PALB2, the VSEP, do not advocate the use of PM4, and that for ATM you can use PM4 for the use of stop-loss variants. Next, please. PM5 is another one where is a big difference. Normally, it originally was described that it should be used for novel missense changes at amino acid residues where a different missense change was determined to be pathogenic and has been seen before. Enigma only allows to apply PM5 for protein truncating variants. And there you can use it at different strengths, ranging from strong to moderate, from strong over moderate to supporting. And you can apply it for exons where a different proven pathogenic protein truncating variant has been seen before. And this can help you because to get truncating variants in BRCA1 and 2 that may not reach the pathogenic criterion because PM2 can only be used at a supporting level, because you have them PVS1 plus PM2 at supporting. It will only lead you to a likely pathogenic variant, but in combination with this PM5, you can most often this upgrade to a pathogenic variant. There is a table 4 where Enigma has indicated per exon on which strength you can apply the PM5, and you can find this in Appendix D of the VSAP guidelines. In contrast, for ATM, you can only use PM5 at the supporting level, and this is for all protein truncating variants upstream of arginine 3047, and for PALB2 only, also PM5 only at the supporting level, but for protein truncating variants upstream of tyrosine 1183. In contrast, the PM5 is used for missense variants according to CanVIG. And there you also have to take into account the prediction scores, and CanVIG is using Revel for the prediction scores, and you can use it at moderate if the variant under assessment has an equivalent or more deleterious Revel score than the reference variant, or both the reference variant and the variant under examination have a Revel scores that are higher than 0.773. You can use it at the supporting level if the reference variant is classified as likely pathogenic variant and has only been reported in one individual, and or if the variant under assessment has a Revel score between 0.7 and 0.773, but less deleterious than the Revel score of the reference variant. The important thing you have to take into account that your variant that you want to classify has also a Revel score that is at least 0.7. Next, please. PS1 can be applied for missense variants with no impact on splicing. So you can use it according to Enigma as strong. For missense variants where previously a pathogenic variant is considered to act via protein change. So you're sure that there is no effect on messenger RNA splicing. So the Splice AI score should be less than 0.1 for the variants. Or at moderate, if the missense substitution was previously classified as likely pathogenic, and you should have excluded that there is no effect on mRNA splicing. And at the supporting level, that was explained by Miranda, it is for exonic and intron variants with a predicted impact on splicing. In CanVIG, you can also use it at strong or moderate. At strong for missense variants or initiation codons, so that's different from the Enigma guidelines because they're so where the variant under assessment is a reference missense or initiation variant and classified as pathogenic, and if it's this reference variant is classified as likely pathogenic, you can only apply it at moderate. And you can also use it supporting, but this is done for the non-canonical splice variants as explained by Miranda. Next, please. Then the codes for the functional testing at PS3 and BS3. In Enigma, you can apply them at different strengths. And I just want to alert you that Enigma has made a very nice overview table. It's table 9, where you can look up individual variants and where they will tell you at which strength you can use it based on the functional assays that have been applied. Please note that for PALB2, there's currently not yet one of the functional assays scored so that you can use them into the for the variant classification, and this is due to the fact that there are no no very well known missense variants that can be used as controls. For ATM, there are ATM-specific assays that can be used at moderate or supporting length. And for the RNA level, you can use the PVS1 strength, as explained by Miranda. For CanVIG, it's quite similar, and they have also a very nice overview of the published functional assays, and you can find it on the Google Drive as indicated here. So they have evaluated the functional assays based on Burnage et al. They consider a variant to be functionally abnormal if the protein activity assay or the functional impact is less than 25% of the wild type level. And they indicate also at which strength you can use it. And theoretically, it can be at very strong, but none of the functional assays are now for none of the functional assays, you can now use it at very strong, but you can apply strong, moderate, and supporting. Next, please. The prediction programs at BP4 and BP4 can also be used, and I wanted to alert you that there might be different programs advised per gene, and there might be different cutoffs per gene. So for instance, here for BRCA1 and 2, biased del, no AF scores can be applied. And you see here that the cutoffs for both BP3 and BP4 are slightly different for BRCA1 versus BRCA2. Another thing I want to alert you is that dependent on the location of the variant, especially for missense variants and silent variants, you can combine PP3 with other criteria. For instance, let's have a look at the silent variants where there is no predicted impact on splicing. Then you have to check, is it inside a functional domain? If this is the case, then you can apply BP4 and BP7. And the functional domains are listed here at the right side of the slide. If it's not inside a functional domain, you can apply BP1 strong. Similar for missense and in-frame variants, for which there is no impact on splicing, also depending on the fact if they are inside or outside a functional domain. So in an inside functional domain, you can apply BP1 at a strong level. And if it's inside the functional domain with an predicted impact on the protein, you can apply PP3 and you may consider PS1, or if there is no predicted impact on the protein, you can apply BP4. And similar for the intronic variants, but that was more the focus of the previous speaker. Next slide, please. So for to continue with the PP3 and BP4, so for Enigma, bias del is used with different cutoffs for BRCA1 and 2. For the CanVIG, there are the REVEL scores. And for PP3, the REVEL should be higher than 0.7, and BP4, the REVEL should be smaller than 0.4. But to make it a bit more complicated, for ATM and PALB2, CanVIG refers to the ClinGen VSEP guidelines. And you see then for the VSEP, ATM and PALB2, for the, determining the effect at the protein level. For PP3, for PALB2, you cannot use, and also not for PP4, just because we have no idea about we don't know about a large number of missense variants that have a pathogenic effect in PALB2. For ATM, it's REVEL that's being applied, but at a different cutoff than the ones proposed by CanVIG. Next slide. Then the BP1 is also an interesting, as I've shown you, you can use it at a strong level for variants that are substitutions, silent substitutions, missense or inframes, insertions, deletion or delins variants outside the potentially clinically important functional domain and no splicing predicted. For BALB2, you can also use it for all missense variants at a supporting level, and for ATM, you cannot apply BP1. According to the CanVIG guidelines, you can only apply BP1 at a supporting level for genes or gene regions in which more than 95% of the reported pathogenic variants are truncating, and as an example, APC and BALB2 are given. Next, please. So to end, I would just like to show some worked examples. I will discuss two variants that were classified in the previous run. Next, please. So the first is this missense variant, and I will illustrate that depending on the guidelines that you are being applied, you can end with a different classification. For instance, this missense variant, according to Enigma, it's a class 3 because PM2 can only be applied at the supporting level. It's not present in the GNOMAD non-cancer subgroup and is also absent from the GNOMAD version 3 non-cancer subgroup. And then you can apply PS3 at a strong level based on two functional assays from Findley et al. And Fernandez et al. Who have a uniform outcome, which you can find in the table 9 of the BRCA1 guidelines. In the CanVIG, it will be classified as a class 4 because you can apply PM2 at moderate strength because it's the variant is not present in GNOMAD version 4.1. And then same argument for the functional assays, you can apply PS3 at strong. Next, please. This is to show you where you can find outcome of functional tests. You can find these in the BRCA exchange. You see here all the papers that have evaluated functionally several variants. And it's also nicely summarized on the CanVIG-UK website, so where you can, from which you can easily retrieve the outcome. Next, please. While several applicants applied some scores that should not be applied. For instance, PP3 could not be applied according to Enigma because it is a Bayes Del score of 0.24, which is not informative according to the Enigma guidelines, and according to CanVIG, it's the Revel, but it's below the 0.7, so the PP3 is not met. PM1 cannot also not be used according to Enigma because it should be considered as a component of the bioinformatic analysis, and according to CanVIG because functional data have been applied, so you cannot combine it to avoid double counting. And there is a nice table in the CanVIG guidelines indicating in a very visual way which scores cannot be combined. And then PM5 was applied. So in Enigma, you can only use this for protein truncation variants in an exon where a different proven pathogenic protein truncating variant was seen before. And in CanVIG, the reference variant, which is a substitution G2T instead of G2C, which results in another amino acid, a valine instead of an alanine. The Revel score is 0.758, and that was classified as pathogenic using the multifactorial likelihood data published by Caputo et al. However, the variant under assessment has a Revel score that's below 0.7, so therefore you could not apply the PM5. Next, please. Then the other variants, the other missense variants in BRCA1, there were similar criteria that were applied by Enigma and CanVIG, but at differing strands. So BS1 can be applied. The original interpretation was allele frequency is greater than expected for the disorder. According to Enigma, you can apply it at the supporting level because the filter allele frequency is larger than 0.02%, but lower than 0.01% in GNOMAT 2.1 and GNOMAT 3.1, both the non-cancer subsets. And I will show in the next slide how you can look up the filter allele frequency. And then in CanVIG, you can use BS1 at a strong level. Because it has a maximum tolerated reference allele count of 55, which is lower than the number of alleles observed in the UK biobank if you consider only the female-only data. I will not go in detail how you can calculate that, but you can find a nice webinar from Miranda on this link that I'm showing here, where she clearly explains how to apply BA1 and BS1 according to the CanVIG guidelines. Next slide. So another criteria that could be applied for this variant is BS2. A BS2 is if you observe it, it's in a healthy individual for a recessive disorder with full penetrance expected at early age. For Enigma, you can only use it at moderate strength, and that is because a homozygous variant was found in the GNOMAT 4.1, the UK biobank data, and it's in an affected individual that's unaffected individual aged between 65 and 70 years. And then the BP1 for CanVIG, you can use it at strong, and it's based on the same arguments. And then the BP1, you can use it according to Enigma as a strong argument because it is not predicted to impact splicing and outside of the critical domains. And for genes, for CanVIG, you can only use it at a supporting level because for genes and regions in which more than 95% of the reported pathogenic variants are truncating, you should use it at supporting. Next, please. So here you can see where you can identify in GNOMAT the filter allele frequency. It's on the top of the page. And then if you scroll a bit down, you can also find the age of the carriers in GNOMAT. And you can also click on on the homozygous variant carriers, and then you see that this was one person aged between 65 and 70. Next, please. So then I would like to end with a take-home message, very simple. So I think I have shown you that different variant classification guidelines may lead to different conclusions. Please know that variant classification guidelines are still evolving. So therefore, it is important to indicate on your reports which guidelines you have used, and not only which guidelines, but also the version, because things are still being followed up and further improved. And with this, I would like to end my part of the talk, and I would like to hand over again to Sandi.
Thank you, Kathleen, very much indeed, and Miranda and Alice for some really, really educational stuff there and a huge amount of information. So thank you very much indeed. Tackling all the really challenging parts of the classification now, aren't we? So just for those of you on the webinar, just to say, if you've not yet enrolled for the next online assessment module, which is run 13, then please do so. Here you've got the link and you can do that. It's free of charge. It's sponsored through an educational grant, and the assessment will be available for for all of the month of October. So you've got a few more days to sign up. We've still got a huge number of people already signed up, so that's great. And as I say, they'll go live next Tuesday. If you want further information, you can contact both EMQN or GenQA here on our contacts. So next slide, please. Simon, are we ready to do some Q&A? I'm very aware that we've not got too much time, but there's some really interesting questions coming in. So just to reiterate, if we don't answer your question today, they will definitely be answered in the EQA summary report from run 13 from the Genie module. So don't worry, you will get your response. But we've got a couple here. Do you want to kick us off, Simon?
Yeah, I will do, Sandi. So thank you very much indeed, Alice, Miranda and Kathleen. A great presentation. Lots of education in there. I've learned a lot as we've gone through it as well. So Alice, I'm going to kick off with you because you were the first speaker, and just a general comment really, more, I suppose, more than anything else. If a user was new to say CanVIG-UK, and many of our participants may well be, where would they go to on the website to find the information about the reduced penetrance guidelines? And I guess, and follow on from that, is there an intention, for example, to publish that in a peer-reviewed journal?
Yeah, so thank you for the question. So yeah, pretty much everything that we've got that's in its kind of final form is on the CanVIG website, and there's different kind of buttons that you click on to access different resources. We have our preprint is actually available for the reduced penetrance framework, and I can put a link to that in the chat. It is hopefully almost at the very final stages of peer review, so we're hoping it will go into press soon, and we were sort of waiting for that before putting the final version of the framework on the website in case it sort of got updated along the way. But I would anticipate it'll probably be on the website in the next few weeks, I would say. So it's not on, you can't find it because it's not actually on the website yet, but it will be under the main consensus.
Okay, that's great.
So Simon, maybe if that's a great timing, because maybe we could pick that up on our webinar in November just to signpost it to people
That's
on
what
the
I was
call.
thinking, Sandi. Yeah, it's a great
that would
idea.
Be really good.
Yeah, fantastic. Thank you, Alice. I've got one more for you. One of the questions that came in. So regarding mild phenotypes, so homozygous evidence, how would you define what a mild phenotype really is?
Yeah, so as I say, in the first instance, we were sort of thinking about BRCA1 and BRCA2. So when we're talking about a mild phenotype in the homozygous form, in the kind of full penetrance scenario, we'll be thinking about a fully blown Fanconi anemia type of presentation. And what a minor version of that would be is going to be quite variable and quite subjective. So you'd need to be talking to your kind of clinical colleagues about whether there are some features of Fanconi anemia, but perhaps not as many as you'd expect to see. A lot of, it's obviously a very, there's lots of different kind of presentations of Fanconi anemia, and people have variable expression of the various different features, but a lot of them do occur usually in childhood, with unusual skeletal features, perhaps skeletal childhood onset cancers, that sort of thing. So it might be that you actually have someone who's been fine throughout childhood, but maybe develops a relatively early onset adult cancer. And then, or it might be someone who hasn't yet developed cancer, but then when you do some sort of laboratory test, there's sort of abnormalities on that. I guess those are some kind of examples that I would think about and it's difficult to be too specific. In the CanVIG guidance for BRCA1 and 2, it says that you need some clinical features and some laboratory features. So it might be that it's just the laboratory features you're seeing.
Okay. Fabulous, thank you. Sandi, I'm going to hand over to you for a few questions.
Lovely, thanks, Simon. So Miranda, here's some for you about your leaky splicing. We always like our leaky splicing. So regarding variants with incomplete or leaky aberrant splicing, so if the defect is around 30%, can you use either PVS1 or BP7, and how do you decide?
So assuming this is for BRCA1 and BRCA2, because that's what the kind of thresholds are, for other genes, it needs defining. But if it was BRCA1 or BRCA2, I think you use your professional scientific judgment. You see where all the other evidence is. If you've got a whole stack of kind of other evidence towards benignity, and this is then supporting that, it's, you know, also based on, was that assay repeated, what's the kind of quantitation of that assay, then you're going to be kind of leaning more towards a BP7 strong. If it's really, if you repeated it, the quantitation is very good and it's really on that borderline, then you're probably going to err with the side of caution and just have PVS1 not applicable. But yeah, it's quite hard to generalize, but you really want to be thinking about that assay design and quality and how good the quantitation is.
And put it in context of the other evidence that you're considering. Yeah,
absolutely. Yeah.
Great, thank you. So there's another one here that's coming just afterwards, again on splice variant. So do you take into account predictions on branch points, ESEs, etc.?
They're basically not very good. So there are a huge number of tools that focus just on those specific sequences, but I don't think any of them are really ready for diagnostic use. Splice AI does kind of claim to look at some of those, and it was definitely trained, because obviously Splice AI is a meta predictor. So as I remember, but please, I may not be right, but I think it was 17 different tools that it was kind of used to train it on, and lots and lots of splice variants. So they did include some ESE and branch sites. So it may pick those up, but they are notoriously more difficult. And we know that, and they're probably the ones that cause a lot more variable splicing, the leaky splicing, and you see the different kind of effects with them compared to the canonical splice.
Probably not yet, then, is the answer.
Yeah, I think as time goes on and we know more, but there are just so many different ESEs in particular, very short sequences. There are so many of them, but hopefully they will get better over time. Yeah.
Great, thank you. And I see, thank you, Alice, you've popped in the chat some links as well, so hopefully everyone can pick those up. Thank you. Simon, back to you. I think we can squeeze in another one.
Yeah, we have. Now, a question is, Kathleen, do we go for a difficult one, or should we go for a difficult one to finish it off? No,
be nice.
I'm going to be nice. No, we're not going to go for a difficult one. I'm going to actually come back to you, Miranda, about Splice AI, because there was a comment here which is quite interesting in terms of the availability of these tools, because obviously the guidelines recommend using specific tools to do predictions, etc. So we're looking at Splice AI for PP3. It's the recommended silicon in silico prediction tool, but it's only licensed for research use only, or non-academic institutions. If you were part of a commercial laboratory, have you got any guidance about what those laboratories may or may not be able to do for supporting this?
I am afraid I don't have we work for the NHS, so it's kind of not, probably not commercial, but I know that all of those are saying for a lot of our lab kind of kits, etc. Then a lot of them still say for research only, and it's just about us doing that appropriate validation. So that's how we get to use it in the clinical diagnostic setting. I'm really sorry, I can't comment.
And I wouldn't expect to do any more, because obviously that is leading on to an IVDR question. We're not going to go there in today's call. That's a subject for a different matter. I will then obviously come on to the last point, which is the learning we've got from the EQA itself from the last round. And Kathleen, there is a question here relating to one of the BRCA variants. I think it was BRCA C5213G2C. Let me just read through. So there's a question really about should one use Enigma guidelines that are should one use Enigma guidelines because they're so strict? The question is really, do you think there's any way around amending that classification? If it said, should I not, the participants, should I say, thought it was a VUS, but obviously clearly there's contradiction in terms of the results from the consequence of that. Do you have any thoughts about I think you gave some examples for that variant, but how many laboratories, for example, gave a discordant result from the expectation?
Yeah, well, it's indeed a hard one, because if you're functional assays are really supporting a pathogenic variant. And because of this PM2 at supporting level, we only end up in a VUS. It will be a hot VUS anyway. So one where there is we just do not reach sufficient statistical evidence to classify it as a likely pathogenic according to Enigma. So, but these are the kind of variants where Enigma is also putting a lot of effort generally. If there are several families where they can which can add additional evidence, like I'm thinking about segregation studies, etc., then it might end up in a higher category. But if at the moment, yeah, these are the guidelines, and if we apply them strictly, we end up in a class 3. And then, yeah, the best thing to do is to try to get it higher by investigating the families where you identify it. Yeah.
I think your summary slide was very clear about that, actually. I mean, it's still evolving, isn't it?
Yeah.
I think this is the key thing we're trying to get across, that this field is not by any means finished. Our ability to analyze sequence data is becoming much more comprehensive, so is the need to be able to interpret those variants, and it's challenging, as I think we could all see from it, but still evolving as a consequence.
Indeed,
Sandi.
and so functional assays are still being set up and might reveal new insights.
Yeah. Sandi, timing wise, how are we doing for timing?
I think we need to close now, Simon. There's a few other questions here, but we can cover them, and some are just comments around, can't we just simplify the guidelines and have one? And yes, life would be so much easier, wouldn't it? But we will answer the outstanding questions as part of the EQA summary report, which will be out after this run that's going live in October. So just finally, just say a big thank you to co-chair Simon and also Alice, Kathleen and Miranda for a fantastic webinar and really, really educational. So thank you very much. I know a lot of work went into that presentation. So thank you again.
Thank you all.
Thanks very much, and we'll see you the next time.